one of the issues that we face basically is that aquaculture is a very diverse sector. So we are working basically with marine species, bivalves, crustaceans, and every single species has its own genome. But at this moment, not all the species actually have the good genomic resources for producing good SNP chips. So I will be talking basically what happened, the history of one of the species that we actually started raising a couple of years back in Chile. Is the yellow-tailed kingfish. It's a highly valuable fish for the aquaculture sector. And it's important because the state and Chile, the Chilean government actually think of changing the paradigm of just using one single species, salmon, and diversify the production to other aquaculture species in an industry scale, you know. So for that, they have actually select 4 species. The Patagonian toothfish is one of them. Maybe you know it's a high valuable fish, difficult to raise, and the biology of the fish is really unknown. They also select the sea bass, the Chilean sea bass, and also the conger eel, and also the Seriola, of course. So we have basically 4 species for diversify the aquaculture sector in the country. And I will be talking about the Seriola, which is the species that we are working with. So many people, when we start working on this and we start the program, many people from the funding sector said, well, why you start working with genomics at the beginning of the program? The thing is that there are many different biological issues for the different species, and they are not resolved very easily. For example, larvae cannot be sexed when they are larvae. So we actually can have populations of unknown sexed individuals. So we can just have a surplus of individuals for giving or having a nice broodstock. So this is one issue. It's not like bovines. Bovines are sexed when they are when they are giving birth, so it's not a problem. The other problem is that because we work with wild species, we actually face another problem. All these species come from a very low number of individuals. So we face the problem of bottlenecks. And bottlenecks are a problem because they actually have an issue about the genetic variability that we are actually using for the program. This is one thing. The other problem is that in the case of Seriola, people raise the Seriola for production. They produce larvae. In fact, the company that is actually working with us, Aquinor, export larvae to Holland, Saudi Arabia, etc. But the problem is that they don't really change the broodstock for a long time. So they have the same broodstock for about 6 years, which is a problem from a point of view of a genetic breeding program, you know. So and this is because the breeding cycle is not really well characterized. For example, there are issues about which individuals actually gave larvae, because the genetic contributions of the parental generation are quite skewed. So you see that, for example, from a spawning, you can have like one male and one female giving larvae to the pool. So it's difficult. And also because we have problems with how the fish are raised. Fish can be raised as in recirculation units, but also in net cages. And the system is completely different. In net cages, for example, the fish is exposed to diseases. Especially in Seriola, one of the most important diseases are the parasites. And this gave a lot of challenges how to deal with these parasites. So all these problems actually can be dealt with genomics, as I will show after. So we're talking about here again about how, for example, these populations actually are structured. When salmonids are not really are alien to the southern hemisphere, so we don't have natural populations there normally. But here we are going to deal with an issue that we have wild populations. We're using these wild populations for aquaculture. So the problem is how we can deal with sustainability of the wild populations. So for that, we need to know exactly what is the structure of the populations that we are working with. Because the reproductive system is unknown, the influence of the parental generation is also unknown, and we have to look, for example, as I told you before, about the parental contributions. As I said before, the sex determination is also unknown, and this is important to actually manage the broodstock. And of course, when you want to apply this in a practical breeding program, we need to know the associations between the SNPs, for example, and the traits. And this is generally information that is not known. And there is no chip available. There is no GTC panel available. We start working with microsatellites, but they are a pain, basically, to work with, especially when you have a lot of samples to do, and the interpretation can be challenging when you don't have microsatellites coming from the same species, the focal species. So it's a problem. I want to just show you what is the distribution of the Seriola yellowtail kingfish populations. We have basically in the southern hemisphere Seriola in South Africa, in Australia, New Zealand, and in Chile. So there is a whole range of different populations in the southern hemisphere that we actually need to survey. In the northern hemisphere, there is Seriola dorsalis that previously was thought to be also Seriola lalandi, but turned out to be a different species. So I will talk about a little more about the diversification of the aquaculture production in the Chilean aquaculture. So this was a program, meaning that it's a long-term program, mid-term program, for developing new industries related to aquaculture in the country, particularly based on marine species. As I said before, we commercialize high-value species. Well, the Patagonian toothfish is one of the most clear examples, with prices, very good prices for exports. And the other thing that the country wants to do is to build partnerships between the public and the private sector. So all these programs actually are public-private partnerships. And because I work in the University of Chile, it's our duty to work as an academic, fulfilling all these duties. So we first developed genomic resources. So the first part of the program was basically develop the genomic resources for the species. We basically develop a draft genome. With that, we use samples from different samples with a certain scheme in order to develop a SNP genotyping chip that basically capture the variation of the Seriola lalandi. We then, with this SNP chip, we validate the polymorphisms in different populations, not only in the Chilean populations, but also in New Zealand and Australian populations. And at the same time, we actually look for phenotypes that were of interest. For example, the deformities. Larvae are prone to be deformed, and we wanted to know exactly what is the genetic mechanisms of these deformities. And of course, the sex determination. We didn't have a very large budget for doing this, so we actually did a scheme that tried to capture both the sex determination and also the variability among the broodstock that we have using for the SNP chip production. But before that, we select one male and one female. From them, we produce mate pair libraries and pair end libraries with a high coverage for each sex. So for that, we actually produce two different genome assemblies, one for the males and one for the females. We use Mazurka, which was one of a very nice programs to do the genome assembly. But the purpose was not to develop a complete draft. The objective was to develop a draft that can be used for SNP discovery. Okay. This was the draft genome. We actually have about 95% of the genome covered with the assembly, and the parameters, as I said before, are not great, but they were usable for the SNP chip discovery. We then sequenced at low coverage 17 females and 17 males to either the male or the female genome assembly. And with that, we discover we have these two purposes with the same data: discover the sex determination region and also discover SNPs. The sex determination in this species is ZWZZ for the males. And we perform a GWAS based on resequence of the individuals that we have before, and using about 500,000 SNPs. Remember, this is only 17 females and 17 males. You see here, this is the GWAS. These are all the scaffolds. But the point here that you need to look at is that one single region basically shows a very specific position for the sex determination. When we look closer and annotate the region, we actually find that there was one gene, a quite interesting gene, because it's a hydroxysteroid 17 beta dehydrogenase gene that actually is determine the transformation of different estrogens to either testosterone or estrogens. When you look at the pattern of variability between the sexes, you see that the females have this pattern of completely heterozygous, and the males are completely homozygous. So if you look at the FST here, the FST was higher in those regions that are actually exons of the gene. We after we have done this, we actually validate the discovery of the sex determination region. Using HRM, we develop a proof that can be used for detecting the sex very easily. We just have to take one sample from the fin or DNA, and then we can actually predict the sex very well. The confidence is about 95%. And this is the gene. You see that it's involved in both pathways. This is the KEGG pathway. And this is the first evidence of a gene known to regulate the steroid production in sex determination. We are actually trying to prove this using functional studies, and we are in the process of developing these assays and test, which is actually going on there. Okay, we developed the SNP chip. We survey about 1.2 million SNPs of high quality out of 5 million, just using the draft sequence. With different iterations, we end up having about 95K SNPs to be included in the SNP chip. And this was a collaboration with Thermo. In the array, we include not only unknown genes, unknown SNPs. Because we have done an RNA-seq analysis, we include SNPs that actually are included in the transcriptome for deformed and normal individuals. We also include the sex determination region, of course, because we need to know exactly what is going on with this region in more detail. So we actually genotype all the SNPs that were in the scaffold where we discovered the sex determination region, the gene. And because we have this collaboration with NOAA, we actually use regions that were homologous to the Seriola dorsalis genome to see if the same SNPs that were variable in the Seriola lalandi were actually variable in the Seriola dorsalis species. So after we had Thermo produced the array, We actually validate the array using biological data. We use populations from either Australia, Ruakaka in New Zealand, broodstock data from the company Equinor, some individuals that came from different natural populations obtained in a bit southern of Caldera, where basically the company is, and some samples from Mexico, which is another source for supposedly Seriola lalandi. We did some analysis based on population assignment using admixture. We found that there is a very distinct pattern between the different populations, but not between New Zealand and Chile. You see that the proportions of the clusters actually are quite similar between Chile and New Zealand. Basically, this is behaving as a single population. Australia, I think, is the same, but the problem with Australia is that we didn't have broodstock data. We have larvae data. So this may be a reflection of the structure within the population that we are actually genotyping. So you see different proportions based on the proportions of the families that were available in the larvae. So it's tricky. But the Mexican population was completely different. We use some demographic analysis with premix in order to test the covariance between the different populations. And this is actually the structure that the program predict. This New Zealand, Chile broodstock, and all the Chilean populations cluster basically together. Australia is a bit farther away, and Mexico was completely out of the other different populations. The variance explained by the relatedness in this graph is about 99%, not including any migration. So it's a very good tree. Because we have a genotype about 400 samples with the genotyping array for the Seriola, we have samples that were phenotyped with the sex. And we have done this in order to test which is the, to validate basically the sex determination region in the samples, in the new set of samples. We have now about 200 individuals that were sexed in our array, in our samples. With that, we perform a GWAS, and if you see, the same region actually holds for the sex determination. This is quite impressive. The only example that you can see in the aquaculture sector is the IPN QTL, which is a single gene explaining the resistance for IPN, which is quite remarkable. So for the implementation, each part, the mating system is quite difficult. You can actually have individuals that are stripped. Like salmon, for example. If you have a female, you're going to strip the eggs, and if you have a male, you're going to strip the sperm, and then you can actually produce your families. Here, the spawnings are spontaneous, so you can actually tell which individual belongs to the different parents. So this gave problems in terms of the genetic contributions, and we just do this based on some samples that we have, and you see that there is a large skewed distribution of the parental contributions of the broodstock. This is using the SNP chip. So this is a problem. The inbreeding of the population of the different samples is quite important. This is the Mexican, this is the Australian, and the rest is quite small. So the strategy of this would be to develop a GTSEC panel in order to first replace completely the microsatellites for parentage analysis. So we can survey a large number of individuals from our population, selecting those individuals that are actually quite diverse, given the parental contribution. And after this, we will apply the genotyping chip for developing the reference population in the Seriola. We are working on that, basically using different approaches for that. I have to say that from the 90K SNPs that we have surveyed, about 85,000 works very well in all the populations, so it was a complete success. The most important trait that we are going to use for selecting the broodstock is harvest, which has actually a quite large heritability of about 0.6. And we have done some small deterministic simulations in order to see what is the actual use of the genomic selection compared to BLAP or basically mass selection. And for that, we use selection, a program comparing mass selection, BLAP, and genomic selection. You have to take into account that it's not possible to have a single breeding population that spawn all in a single space of time. So we have basically 4 different groups that have different thermoperiod and different photoperiod. So throughout the year, these different groups spawn and produce larvae. So from these different reproductive systems, we have to take the larvae for the reference, to take individuals for giving the reference population. So there are 2 possibilities. We can actually select for using the information from within the group, or selecting based on the whole system of reproduction. And the problem with that is that we expect that for the case of the group strategy, within single spawning tanks, we can have a lot of inbreeding. And this is what this is shown in this graph. When you have the inbreeding, for example, it's about double of what you expect given the inbreeding in the entire population. You see some differences in terms of the genomic selection based on the group and the entire population, but the main point is that at the same rate of inbreeding, the response is much larger considering the entire group. The problem is that with BLAP selection, you cannot use BLAP selection based on the whole entire population because you don't have connections between the different spawnings. So this is a problem. So BLAP is not possible to be used. You can use mass selection, but increasing the gain is worth in terms of the inbreeding. So when we start, many people say, why you want to develop genomic resources and blah, blah, blah. But I think it's important when you want to develop a healthy industry to invest in genomic resources. We show that with not a large amount of money, we actually have produced a lot of information that is actually used for the company and the production system. For example, in terms of the sex determination. And also, it's important to realize that when you use this kind of technologies, you're actually using also this information for produce a sustainable breeding program that has less inbreeding and also the larger genetic gains that you can have within the system.